Welcome to Agri Academy. Welcome to Genetics CP. In this video, we're going to discuss about the previous year question which I asked in the 2025. I think it may helpful for your examination in 2026. First question is, in a triple test cross analysis, the L1 plus L2 minus 2 L3 values given resulted in an estimate of minus 12.46. Which of the following statements true with respect to triple test cross analysis and the current results? If you want to understand this question, you should know what is triple test cross analysis. In a simple way, it's an extension of a North Carolina design 3, where in North Carolina design 3 we cross parent and parent 2 and we develop an F2 population. We select randomly some genotypes and we may cross with P1 and as well as P2. And along with, if you cross the F2 for F1 also, then it will become TTC. Means, how you will estimate, for example, L1, L2, and L3. Means P1, P2, P3. The progenies you will get like this. We use this population for estimation of the genetic effects. Then this design is called as triple test cross analysis. And this triple test cross analysis provides either presence or absence of your epistasis. And the equation is L1 plus L2 minus 2L3 is equal to 0. Then only there is a no epistasis. If there any deviation from the value 0, it leads to there is the presence of an epistatic interaction. I hope you understood the question. The statement 1 is correct and the statement 2 is wrong. But many people have kept this statement as correct. Why? Because we are reading wrong information regularly. This information present in Punjan Singh and S.S. Narayan book, where he present the greater than 0 is indicates presence of an epistasis. But what is less than 0? It is a minus 12 like this. What it estimates? Means there is a absence of epistasis. No. The any value deviating from the 0, it is indicates that there is a presence of an epistasis. Okay, we'll move to the next question. Then, this is a very simple topic. For example, you can estimate it in a QTL mapping, the number of detected QTL with their effect in the means of the phenotypic variance explained. The population size is known as the BVS effect. If you read this paper, I think you'll get, I think it's not a original paper, but it's giving a basis for the BVS effect. See, for example, you can read the sentence clearly here. If you use 100 progeny, the QTL estimates are slightly overestimated. For example, you are using 100 individuals, you get some QTL. You can take 30 PV, 80 phenotypic variance explained, sometimes 50 phenotypic variance explained. It's overestimation. If you use the same marker with 1000 individuals, you may get around 20% PVV, or it may be, may not be. It's an arbitrary values. But the core concept is if you use the more number of individuals in the mapping population, then your precision of QTL mapping will increase. Okay, that is called as the BVS effect. Then, and here also it's a very simple question. It's a center of origin question. And here you have to know one concept called as old world, and new world crop. And which crops are belongs to old world and which crops are belongs to new world. If you want, you can read this paper, Works 2012 et al. How an apple and an amaranthus, cashew, ananacho, what tobacco, lima bean had moved from a new world to old world. And some crops move from old world to new world. And you should also know which crops are moved interchange between them. For example, the crops like bajra from Asia wheat has moved to the American continent, and some crops like peanut, maize, vanilla, pineapple, it is moved from South American center to the African center. This you should know like clearly. Then there is another question they were asked about in the match the following: one factor, two factor, three factor, four factor belongs to which? It is simple. Simple trick if you want to remember this. See which of your design or analysis will give least information. Put them in factor one and next list and further next list. Like that, you have to arrange and you have to just guess it if you don't know means. If you are reading Biometrical Techniques by J.R. Sharma book, this you have a clear table in that book, you can easily get that. Then, if you are like top cross, it will give only information about GCA. You are line into tester, it will give GCA and SAA. Means you get additive and dominance. And quadrallele, it also gives an epistaticus. Sorry, and it also gives epistaticus. And here you have to make the crosses differently. Here you have to make the based on the complexity, the factor will increases. So you can easily guess based on this information. Then endonuclease and exonuclease are involved in the generation of major matured eukaryotic rRNA cleavage and trimming precursor of RNA. I think these genetics, and I refer you to for genetics, read book your Mustard and Simmons. The questions are always the questions are copy pasted from the Mustard and Simmons book because it's as of now it's a very standard and easy book for genetics. Okay. That's why whenever you are reading genetics, genetics made easy and Mustard and Simmons are very, very important than BDC book. Not only this information, there is another information that I will going to explain in next coming slide. Then this is also, which I already discussed, read plant immunity review paper present in the Nature by Jonathan Jonathan and Dangle paper. Here it is a mechanism of a jet scheme of immunity. For example, when the pathogen attacks, when the pathogen attacks, it having some patterns. For example, some flagellas, whatever it may be, some pattern, it's recognized by the host. Then there is a pattern triggered immunity. Okay. For example, if you want to explain, I will explain now here only. For example, if the pathogen patterns were recognized, then pattern triggered immunity. Host will recognize the patterns of the pathogen, then there is the immunity in the host. So there is increase in the immunity. But the pathogen produces the effectors. Then there is a decrease in the immunity. Then there is a then R genes produced by the host, then this R gene provide the immunity. This jet scheme is called plant immunity, jet scheme of plant immunity presented by Jonathan and Dangle paper in 2007, I think. It's very, very important paper. Please, everyone should read. And there are some specific pattern recognition factors present in the host and some receptors which are placed to effector recognition, like R genes. Okay, and there are God hypotheses. There are several hypotheses present. Please read this paper, it's very, very important. And also remember classical examples which are present for pattern triggered immunity. Your EFR, EFTU receptor is for the pattern triggered immunity. It's not an effector triggered immunity. Okay, then these are the like what is pattern triggered receptors, what is pathogen associated molecular pattern, what is MAPP, what is effector triggered immunity, what is damage associated molecular pattern. For example, if a nematode infecting the rice, what it will do in the root, it damages the root system and it enters. Means this damage is recognized by the root, then it will then damage triggered immunity. Okay, it is also called as herbivore and host interaction. If you have a time, like now you don't have time, I know, but for studying these things, pathogen host interaction, read your one physiology book is called Thomas and Zeiger book, Tom and Zeiger book something. There this chapter is very important. For if any next juniors are reading, preparing for SRF, you just read this book for this particular chapter, host pathogen interaction. And you should also remember the PR classification, PR protein classification, because last time there are one question called as LR34 is having this type of protein. So like ABA protein, because every PR protein have some signature sequences. For example, here leucine rich repeat. But not all PR protein having LRR repeat. Some are peculiar in nature. Some having only some different patterns. Some are having hormones in their nature. You should read. There is a 9 or 7 classification of PR proteins. You must and should remember those molecular nature of those PR proteins. Then I think this is a very important aspect about speciation. And I already discussed you have to read the speciation concept before going to the exam. See, there are different types of speciation allopatric, peripatric, parapatric, and sympatric. For allopatric, just now for the time being, I'll discuss only this. Is there any barrier that stops the reproduction between the two subgroups of a single group? Then, if there is no mating due to any physical barrier, they will eventually become a two different population or two different species. This is called as a allopatric species. Let's see the question. See, geographical isolation is essential for allopatric speciation. Yes, because it stops the reproduction between the two groups, so it will become a different species. And sympatric speciation means sympatric speciation means due to several other factors without any physical barrier, um, there is a subgroup within the group. Okay. If you want to read, I already told the if you read Pranav book, nice examples are present here. You can read that paper. If you read that chapter, you can easily guess this question. Okay. Then then this is present directly in the BD Singh book. No need to explain. Then here, for example, you think there is a marker M1, M2, M3, and M4, M5, M6, and M7. See, they are not talking about M1 and M7 because they are only each error in non-terminal marker causes two apparent recombinant. Means they are talking about non-terminal marker. Means these are M1 and M7 are terminal. They are just talking about these non-terminal markers. They are telling if, for example, if M3 marker and M3 marker, there are two particular alleles, capital A and small a. And in one genotype, you are scoring capital A should be there, but by some mistake, your some band is not coming like that. You can think you are scoring it as a. If you are scoring this wrongly genotyping this marker, means this side and this side, there is a. You can think there is no recombination. You think, okay, then this entire distance will be inflated. Here, think 1 centimorgan, here 1 centimorgan. If you wrongly genotype, definitely the two times there will be increase in the distance. So that's why this both the statements are true. If it is and it is not applies to the terminal marker because this side no marker and this side no marker. Okay, only non-terminal marker having two sides. Okay. I think you understood this question. Then the honeycomb designs. See, everyone know about what is grid designs, what is honeycomb designs, rhombic designs. Okay, these are the designs where they are called as non-replicated designs. For example, in early generation trials, for example, you have to screen 2000 F2 segregating population. You can't replicate it. Okay. For example, you take 400, 4000 lines in F3 or F4. Now we have a different families also. Okay, in pedigree map, you can one family, second family, third family, fourth family. So you can't replicate your 4,000 individuals. So what you can apply any of these designs to select better to alleviate some competitive effect or environmental effect, whatever it may be. So these are, and there are two questions this time. Okay, this design and they are efficiently used in early generation trials. Okay, here you can see all it will be correct, but you can see the particular C option, allow selection only. See, in for example, if there is a different families in the F4 or F3 divided F4, if you are selecting only within the family, no, you are selecting among the families also. That's why this statement is wrong. Then this is very easy question. Those who are very much well aware about protein estimation by Kejel-Dahl method, they are very much aware of this one, actually 16%. Then methods for off early generation test. I already discussed in previous slides. Then here again the question comes. Precision QTL mapping. See, the word precision means you are directly reaching to the your gene of your region of interest, QTL region. Means if you are getting this precision means you are narrowed, should have a very narrow window. This marker 1 and marker 2 interval it should be very narrow. If it is very far away, marker and 1 and marker 2, there are so many large chunk of the region will be there. So among this, which is your gene of, which is the region which are more uh, associated with your trait of interest? So if this precision should be achieved means you should have very narrow interval between the markers. This is called as confidence interval. Okay, means it is not directly proportional, it should be indirectly proportional to the size of the confidence interval. And the rest other is true. Then this is again a glycemic index based question. So if you're reading a paper published by IRRI about glycemic index, low glycemic index, it's mainly important for diabetes 2 patients. If you read that paper, you will easily get it's less than 45 or less than. It means ultra low GA. Right. And you should also remember which is the land races having high, low GI, high protein, high iron and zinc. There are kalanamak rice having high anthracene content. Like this, you have to remember land race and their economic importance and the important quality traits. Then, PPFRA 2001 gives the protection of field crop varieties for the period of. See, this is very important, very easy question. That's no need to discuss. See, here also there is a question based on the sequencing. Polymerase based a real time sequencing. See, the SMART, single molecular real time sequencing, the name itself tells. Okay. Rest other, they are all NGS techniques, but they are not do real time sequencing. Okay, there is a separate process. You have to construct the libraries, then you have to sequence the libraries, then you have to assembly. There are a lot of things are there. Okay. And please read what are the first generation techniques, second generation techniques, third generation techniques, second generation or next generation techniques. Okay, third generation techniques. What is the difference between them? How third generation and second, if you combine next generation and third generation, nowadays are people going hybrid sequencing to improve the accuracy as well as your improve the cost also, reduce the cost also. Okay, these things you should be aware of before going to the exam. Then this is again a very easy question, directly taken from the Mustard and Simmons book. Then this is also again taken from Mustard and Simmons book. And this is common question that like in every exam there is a based questions based on the genome editing. You should know what is SNT, SDN2, what is SDN1, SDN2, and SDN3. I think those who are preparing seriously for the exam, they already know that I don't want to waste the time of them. Then, this is, for example, if you know about this question, you answer. If you don't know about this question, anything, don't try to guess. Because even I tried to guess in the last year, I made a mistake. Even though I was very well aware about what is paternal half-life, what is maternal half-life, what is the importance of in developing double haploid lines, and which are the sources of paternal half-life, which is the sources of maternal half-life. But even though there are classical QTLs are present, but who will ask? Exactly. See, now paternal haploid generally they are not used at all. And going in depth to do like which is the gene and which is the enzyme it produces, it is to make you to confuse you. But better if you don't know, don't ask this question. Then it's a very simple question. Those practically important. See, in a Manhattan plot, if you see x-axis will be the chromosome and y-axis will be negative logarithm of probability, p-values. Okay, it is not negative logarithm of odds. Both statements were wrong, and first statement is correct. Then again, it's a very tricky questions, and most importantly, repeating from year to year. See, if you want to know this concept, you should read biometrical basis of heterosis from B.K. Mukherjee book. It's very interesting book and very small book. I think we can finish in one day itself. Okay. See, what is D, what is Y square? I think those who are preparing seriously, they already know, but I'm telling those who are just watching for the next year students. See, D means degree of dominance, and Y is the allelic difference between the two parents. For example, they're clearly telling they're a double haploid, means either the A or small a, means they are different allele or same allele, means they're completely homozygous in nature. If you need to maximize the heterosis, see, they're asking maximizing the heterosis. See, why take the value from 0 to 1? Okay, 1 which makes you the maximum heterosis. D 1 square means definitely it increases the heterosis. If it is 0, then definitely if it makes the total value 0, then heterosis will become 0. Means the y is equal to P1 minus P2. It's the allelic difference between the two. For example, you substitute 0 and one parent and another one. You will get minus 1. 1 minus 0, you will get plus 1. You square it, both will become 1. So in this case only, you will get maximum heterosis. If you substitute both intermediate values, then definitely if you 0 to 1 you take, definitely it will take 0.99 also, 0.01 also. Then it is less than your 1 only. Okay. That's why your the answer is 0 or 1. There is another concept. There is another concept. It's called as degree of association of the gene with the like effect. Means RD. So RD should be less than 1 or equal to 0 to get maximum heterosis. And there is a two terminologies, complete dispersion and your extreme dispersion. Complete dispersion is better for your getting maximum heterosis, where extreme dispersion is not good. What is complete dispersion? What is extreme dispersion? I already explained in my class of heterosis. Please watch the video present in my YouTube channel. Then, see here, they were asking about genetic determination. Those who are reading Faulkner book, they can easily catch this word and answer. It is means the broad sense retireability itself. F2 minus F1 divided by F2 is going to get the broad sense retireability. The answer is with you. Then the CRISPR-Cas9 system, the role of scaffold region. See how tricky it was. Generally, we forget this region. Okay, many of you don't know, not even study what is the structure of single-guide RNA. That is pathetic. Then you can see this. This is actually the structure of your Cas9 complex. See, these are your Cas9 protein, where this is the structure of the guide RNA. This is the scaffold region, it is associated with the Cas9 protein. It activates it actually. Okay, it's associated with and activates the Cas9. Even though if you don't know this concept, you just think about remove the off-target edits, cleavage of target DNA sequence by itself, pair with the spacer region. By looking at the option, you can eliminate, and only it will be the positive answer. Then, and this is also very simple. Many people miss this in the examination. See, they are asking the duplex. What is duplex? The minimum two capital alleles. Okay. Then, see, what is the ratio of this combination? See, for example, You take this is A1 and this is A2 and this small a1 and small a2. A1 can combine with A1 and A2 and small a2 can combine with small a1 and A2. There are four combinations of capital A and small a. Okay. Then rest only one, four, one. This you can read from PK Gupta book. And there is another possible hot topic is your double reduction. So the name itself tells reduction double time. What is reductional division that you all know? It's meiosis. See, for example, if it is only diploid, for example, if there is a AA is there, and the possibility of getting AA gamete is very, very less, and it's not at all possible at all. But in case of if you are having a auto tetraploid condition, this is a triplets, the possibility of getting small a gamete is there. You can see it is separates, you will get small a gamete. This is called as double reduction. And the alpha, what is alpha value and how to estimate it and what are the four conditions that makes and four conditions must satisfy to double reduction should happen. And these 4 points are very important for the examination. Read PK Gupta for this information. And it is directly present in the APSing and BDSing. There is no explanation for this. It's a memory based question. Then there is another important directly present about functional marker. See, there are three classes: random markers, functional markers, and genetic marker. And if you want detailed information for this, you can refer my marker chapter present in my YouTube channel. And all the information that are freely available. Okay. Then we can just see functional markers are derived from polymorphic sites within the gene affecting the phenotypic variants. Definitely, if it is not affecting the phenotypic variants, then it becomes a genetic marker. Okay, within the gene, but not associated with any phenotypic variation. If it is associated, it will become functional. You just see function; it's associated with the function. If it is not associated with the function, then definitely it will be a genotypic marker. Then if it is not in the gene, not associated with any function, then it is called as random. And many people can ask, even it is not in the genic region, but may affect the function. That also possible. But what kind of marker it is? I don't know. Then, which of the following multivariate analysis? See, multivariate means it can analyze multiple variables at the time. There are two types, like interdependent and dependent multivariate analysis that you can read from a Venkat Reddy book. It's clearly explained what is those two classifications. But here there is a if there is a lot of missing data, you cannot use PCA actually. You can use principal coordinate analysis. If you want in detail about read Ekesing and Bidising book 11th chapter, or you can read there is a one classical paper present in crop science, I think, by Mohammadi et al. Genetic diversity approaches in plant breeding. I think the paper name is Mohammadi et al. 2003. It is clearly present these type of information there. Then linkage disequilibrium information. This is also a very straight question. But how it but the if you want in deep in detail information means, for example, how LD is useful in MTA, you should question yourself. And how LD is helpful in the population genetics in which aspect, and analysis of domestication, how LD helps in the domestication pattern. I think many of you know selective sweep mechanism, selective sweeps, selection signatures. Okay, now at this time you don't read these things, but if next year if you are preparing, anyone preparing, to read these things. What is demographic history of plant? Like what I said, selection, signatures. LD is very, very important to study all of these. Then how it is? You read this paper. You read one important paper published in Nature, I think, Nature Genetics. I will share the paper in my WhatsApp group. Then, Malki, this is again a very important question published in Nature. It's a facts-based question. Then, This is again, it's present in the your Nestor and Simmons book. I will not go in detail. This is also present in any common books. Then the smallest F2 sites having eight segregating and seven non-segregating gene pair. See, don't worry about non-segregating genes. Just worry about eight segregating. What is the population? 4 power n. Then definitely you'll get the answer. Then, see which of the following multivariate analysis with respect to categories. This is also present again, directly present in the Venkat Reddy book also given, Keynotes on Genetics, or you can directly refer Biometrical Techniques by J.R. Sharma book. Tables are direct. And I repeatedly observed from 2025 and 24, the questions directly copy pasted from this book, Biometrical Techniques by J.R. Sharma. Directly putting from just cutting there and pasting here actually in the paper. Okay, if you read this book, definitely you're going to get some marks. Then this question is very, very tricky and very simple also. See, the selection intensity, they are talking about selection intensity of a trait A and trait B are 0.05 and zero point 0.1 respectively. Suppose if the geneticist wants to select both the traits simultaneously, they were talking about the simultaneous selection of both the traits. You should know what is the different methods to select independent culling, selection indices. Okay, there are three different methods. Okay, and tandem selection. There are three different methods which clearly explained in the Dobelker book. Please refer that book for reading this chapter, simultaneous selection. But as of now, we will come to this question. What would be the probable better selection intensities you should employ trait A, trait B? See, you just remember if you want to select two traits simultaneously, then definitely you have to compromise with the selection intensities that you all know. For example, if you are selecting 0.05 means out of 100 you are selecting 5 plants for a trait 1. Here out of 100 you are selecting 10 plants. Sorry, what is 0.1? 10 plants from 100 plants separately. But you're combinedly selecting this trait mean definitely the intensities will be less than this. Sorry, more than the intensity has to be compromised. So you just check where this is possible. Only this here possible. See here the stringency is more here 0.02 and 0.05. It is not the answer because you are selecting very less plants here. But if you are combinedly selecting two traits together, you have to compromise with your intensity, means you have to increase the number of plants. Okay, then here also you see 0.05, it's not be the same. And here 0.001 is further less among 1001 plant. If you are selecting one plant, this is that's why the selection intensity should be compromised when you are doing simultaneous selection. But simple, these are the values which are less than here. That's why uh, more than this one. That's why you have to select. And I think it's more complicated because if you don't know what is I, okay, how it is estimated, what is the meaning of it, if you don't know this, you're not going to understand this. Then the sweet tea. It does not means anything related to sugar. Yeah, it's somewhat related to sugar, but it is more related to the your defense mechanism. See, they're having tail factor, talent, sorry, tail factors tail effectors that can induce you. That's very important in the host recognition by the pathogen. So the sweet genes are the part of the family sugar transporter in the plant. It is related to sugar, but this the tail factor present in this transporter is recognized by the pathogen, then it leads to susceptibility of the host. Please read this paper and how your resistant genes for bacterial blight is related to this mechanism. It's very important. Then, the ratio of number of null hypotheses wrongly rejected to the number of null hypotheses rejected in the experiment. It is called discovery rate. And this is directly present in the Ekising and Bidising book. I'm not going in detail. But you should know why we are using this. In the GWAS analysis, genome-wide association analysis, we are dealing with, for example, you take 50,000 SNP marker. See, the number of testing you are using. For each marker, you are deriving the hypothesis. This marker is associated with your trait, and the alternate hypothesis, this marker is not associated with your trait. Sorry, associated with trait is alternate and not associated with your null hypothesis. Means for every marker, you are driving an hypothesis. And definitely, if you are proving means there is a, you are accepting that there will be 0.01% error or 0.05% error. For your 50,000 markers are there. See how many times into 0.05 only you think. Two zeros means 5,000 false associations you are accepting yourself. Okay. Then what is the meaning of this? Okay. That's why you have to adjust the p-value. If you not directly use the p-values, you should adjust the p-values. So you have to use some correction factors. So these are the FDR. There are Benjaminic correction factors. See, what the problem is called as multiple testing problem. Okay. Oh, deleted. Okay, that's why you are using some Benjamini correction factors, fault discovery rate. There are different factors are there for correcting this problem. We just read from AKC and BDC, which is fully explained. Then, this is again a very simple question. The one trick which I am going to tell you, if there is a 13 resistant and 3 susceptible, and a 9 resistant and 7 susceptible, and there is this particular ratio will be there. Then here one recessive, one this one, two recessive genes are governing the resistance. There is a one mechanism I will provide this table for you. If you just remember this tricks, you're going to get. Otherwise, you have to do all the four possibilities: R1, R2, R3, R4, and two are recessive, two are dominant, or one recessive, one recessive. These two combination have to make. But if you remember 13 is to 3 means one recessive and one domination. If it is 15 is to 1, two dominant R genes are governing the resistance. If it is 9 resistant and 7 susceptible, then two recessive genes are governing your disease reaction. Okay, for example, you think this is in F2. Here in F2 population, 384 among 384, 296 were resistant and 88 susceptible. You just 296 and 88, you just check which is the ratio. If you are getting 13 is to 3, then definitely one dominant and one recessive gene in independent action will governing the resistance. This one, gene action. Then if you are getting 15 is to 1, two dominant and independent, and if it is two recessive, means you are getting 9 is to 7. But there is another thing. These are all independent. But when there is a linkage, there is again a several pattern that I have discussed in my slides. In I think in previous videos I have discussed in disease resistance breeding. It's present in my YouTube channel. You can watch this. Then this is again directly taken from the book Post Translation Modification in the chapter of BDC. If you read this chapter, you will going to get an answer. There are so many signature sequence, pre-sequence, NLS sequence, which is all fold translation modifications. Then NAS pistolate, this is also present in directly. See, one trick you have to remember. Generally, we think that if it is above more the temperature, higher will be the sterility. But in the case here, male fertility will be in the more temperature, above temperature. Okay, means if you have to maintain the male lines, then definitely you have to go for fertile lines. You have to grow them in the above 31-degree temperature. Then, if the type of apomix is the nucleus of the egg cell and genetic cell divide independently gives rise to hybrid camera. Then, with further develop into double haploid in cotton. See, in cotton means semigamy. See, in semigamy, what happens? If you read Chahal and Ghoshal book, you will clearly able to understand. There is a fusion of male and female gamete, but male and female independently develop into the haploid gametes, and they will produce haploid plants. That That procedure is called as semiconductor. Then genetic constitution where this produces phenotypic class where both dominant allele present together. It is very easy question, direct question. Then the progressive heterosis which I told yesterday's class where double cross hybrids are more heterotic than the single cross hybrids. Okay. For example, you take, I think last class I explained, you watched my previous videos like important topic 2 video, we're going to get this point. And here, actually, I was also confused. I kept both are true because it's generally applied in autopolyploid. It's never going to happen in allopolyploid condition. That I was clear. But there is a method only. In biology, there is no word called as only. There is always an exception. There is an auto-allopolyploids also there. In this condition, also your progressive heterosis will happen. Okay. Then breeding behavior of monosomic wheat that is present directly in the table form in BDC group to directly refer this. Then it's also a very very simple question. The answer is 1 by 6. How I will tell? See, neck color of the bird controlled by a pair of codominant alleles. See the word codominant allele such a way that NRR produces the red and white produces the white color. And the heterozygous produces the different peanut pink color. This is clear. Then again, there is another locus which are independently secreted, it's not linked. Then FF will produces the normal feathers, whereas heterozygote will produces the low feather, but another homozygous will is lethal. Then what is the frequency of pink-necked and normal feather birds if dihybrid pink-necked and glow feather birds are crossed? See, dihybrid pink-necked means heterozygote and glow feather means heterozygote. Both are heterozygote. For example, NR, NW, how it is segregates NR, 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 NW, NR, sorry, NW, NW. In which ratio? 1 is to 2 is to 1 ratio. Okay, 2 by 4. This is clear. Means you have, if you, for example, what they are asking, it's the pink necked and normal feather. Pink necked. You want pink necked means you will get 2 by 4. That is you are going to get. Then pink neck. And another one is normal feather. Here you go. Normal feather means what is the FGF? Same again, it will segregate in 1 is to 2 is to 1 ratio. See 1 by 4 and 2 by 4 and your 1 by 4. This is completely died because of this. Then you think 1 by 4 out of 1 by 4 plus 2 by 4. 1 by 4 plus 2 by 4. You just solve this. 1 by 4, what is this? Okay, 3 by 4. 1 by 4 divided by 3 by 4. Okay, 1 by 4 divided by 3 by 4. It's 12 by 4, I think. Sorry. You just solve it. I'm very bad in mathematics. We get going to the 1 by 6 answer. Then, among the given statement about pseudogenes, it's present directly in the Nusted and Simmons book. No need to even read also, or even discuss also. Then, central limit theorem. This is very, very, very easy question. There is, if you read basic statistics books, any basic statistics books, you're going to answer this question. If you don't know answer, you just guess. See, normally what will happen if anything you should have in independent random quantities. If wherever the non-random, you just cancel those options. Okay. Then the sum of the large number of independent random quantities always diverges from normal distribution. It's not that. The theory was if there is a normal distribution. To take random sampling from this distribution, it also gives the normal distribution. This is based on this theory only, where all the whatever the analysis we are doing, it's based on this assumption actually. The normality means if you're sampling from the large data set, random sampling that is following normal distribution. That sample also follow the normal distribution. This is only central limit theorem. If you know that concept, you can easily answer. And this is again a very simple question, but it's a factual question actually. Those who are taught in the class are they are lucky to answer this. Otherwise, it's very difficult to answer. Okay, this type of question you don't even touch. You'll get the negative marks. Okay, with this I complete my session. All the best for your examination. If you have any doubts, you can reach me at any time through my WhatsApp group. Okay, bye.